Chapter 4, Energy and Cellular Metabolism. So in this chapter, we're going to talk about energy, different chemical reactions. We'll look at the role of enzymes in your body, and then we'll end with making ATP and making new proteins. So energy is required to do work. There are three types of work in your body. We have chemical, transport, and mechanical work. Chemical work is the work of making and breaking chemical bonds, the same types of chemical bonds we talked about in Chapter 2. Transport work is the movement, the transport of ions, molecules, and other particles through your cell membrane. And by doing that, we can create concentration gradients. And this is where you have a difference in concentration across the membrane, typically. Sodium, let's use that as an example. We have a higher concentration of sodium extracellularly compared to intracellularly. And by moving sodium out of a cell, we create this concentration gradient. The last type of work is mechanical work. This is the work of movement. So organelles moving around inside a cell. We talked about microtubules acting kind of like a railroad track. We have cilia beating, flagella beating, muscles contracting. So all of those would be part of mechanical work. Now, energy comes in two forms. We have kinetic and potential. Kinetic is energy in motion. Potential energy is stored energy. So this man pushing this stone up the ramp is an example of kinetic energy because he's moving. Now that the stone is at the top of the ramp, he has stored potential energy. So this stone has the potential to roll down, and it does. So then that potential energy is then converted back to kinetic. So energy can be converted from one form to another, as seen in this example. Now, how does that apply in our bodies? Well, again, we're going to go back to concentration gradients across the cell membrane. So I'm going to use the sodium-potassium pump as an example, and that's a pump you'll become familiar with in coming chapters. Let's focus on sodium, part of that sodium-potassium pump. The pump pumps 3 sodiums out of a cell. And as it's pumping sodium out of a cell, that would be an example of kinetic energy because we have movement happening. And we're going to continue to pump sodium out of a cell, creating a concentration gradient. There's going to be a higher concentration of sodium extracellularly, a lower concentration of sodium intracellularly. Now, you're going to learn in coming chapters that ions want to move down a concentration gradient, just like that stone wants to roll down the ramp. So then that potential energy we have created by moving all of that sodium out of a cell will then convert back to kinetic energy, with that sodium then rushing back into the cell when a channel opens. So a lot of these terms you may not be familiar with, but you will become familiar with it as we continue on. Okay, so let's move on to chemical reactions. For chemical reactions to take place, a lot of the times an activation energy is required. And simply put, it's the energy that we have to put in to activate that reaction. So if you look at this man, he wants to take this big old stone, which we're going to call the reactants, and turn it into little pebbles, shown here as products. In order to do that, he first has to put in some energy. He has to push this stone up the hill first. So that's the amount of energy he has to put in to activate the reaction. Reactions can have low or high activation energies. If it has a low activation energy, it means the amount of energy you need to put in is very small. So these reactions tend to happen more spontaneously. Reactions that have a high activation energy, you have to put in a lot of energy for it to happen. That means that reaction is less likely to occur, so the reactions are typically slower. So if we were to move this man down here, and now he has to push the stone from way down here all the way up the hill, that would be an example of a higher activation energy, and so he's less likely to be successful at getting this reaction to occur. Reaction rate is just the speed of the reaction. So another thing I want to talk about is coupling of chemical reactions. So in certain types of reactions, when the reaction takes place, energy is created and released. So here A and B turns into C and D, and when it does, energy is released. This is an example of an exergonic reaction. Other types of reactions require energy. We need to put in energy for the reaction to take place. That would be an example of an endergonic reaction. So what we can do is we can couple an exergonic reaction with an endergonic reaction. And that's where ATP comes in. That's the energy of our body. So what ATP does is it turns into ADP. And when ATP becomes ADP, now the T means try, the D means die. And remember, from previous chapters, we learned that energy is stored in chemical bonds. So when ATP triphosphate loses one of those phosphates, a bond is broken, and that breaking of the bond releases energy. So that exergonic reaction will provide the energy required for other endergonic reactions in our body to occur. So that is coupling. Next, enzymes. Enzymes, I'm sure you've heard about. We know they're in our body. What do? Well, they speed up the rate of reactions, and they do that by lowering the activation energy required for a reaction to take place. And really, this is accomplished by putting the substrates into a more favorable position for the reaction to take place. So essentially, an enzyme will take this big old activation energy and lower it. And again, we know that lower activation energy reactions are more likely to occur. Enzymes are also biological catalysts, which means they don't change when they are used, so we can use them again. And most enzymes come in the form of proteins. So let's take a look at different reactions in our body. We have catabolic and anabolic. Catabolic reactions produce energy through breakdown of biomolecules. So you're going to take something big and break it down. Anabolic reactions utilize energy to synthesize and create larger biomolecules. So anabolic is the opposite. It takes little things and it creates something big. We're going to then move on to the examples of ATP production and protein synthesis. So follow along with ATP production. This is a good overview slide of it, and then we have some breakdowns coming up. So there are two major ways we make ATP in our body. Again, ATP is that energy that we want to fuel different reactions in our body. The first way that we make ATP is through aerobic metabolism, and that's really shown here. Aerobic metabolism is called aerobic really because we use air or oxygen. And by doing aerobic metabolism, it actually yields the most ATP. So it occurs in cells, and here all we're seeing is the cytosol of a cell and the mitochondria because these are the only parts of the cell that are really required for ATP production. So the reaction takes place beginning in the cytosol, and then it will move into the mitochondria. So let's start from the top. We make ATP by using glucose. We take glucose, C6H12O6, and we basically make some changes to it. And we, change it a little bit, add something to it, break it apart, etc. All of those little changes, we just give it a name, and we call it glycolysis. So glucose will go through a process called glycolysis. And while it's going through the process called glycolysis, we make some ATP. And at the end, we end up with two pyruvates. We started with one glucose. We're going to end up with some energy and two pyruvates. Now, in the presence of oxygen, the two pyruvates will move into the mitochondria. Now, remember, the mitochondria has a double membrane. We're actually going to move into the inside of the mitochondria. So inside the inner membrane. And the two pyruvates, once it gets in there, will turn into acetyl-CoA. So there were two pyruvates, so you'll get two acetyl-CoAs. And you'll make some energy. The acetyl-CoAs will then attach to another molecule, and then it'll go through a series of changes. You don't need to know all of those changes. All you need to know is that that process is called the citric acid cycle, also known as the Krebs cycle. Krebs is in K-R-E-B. And in the process of acetyl-CoA going through the Krebs cycle or citric acid cycle, you make more energy. Then what we're going to do is we're going to take the energy that we created. Some of that energy is going to be in the form of ATP as shown here. Others are going to be in the form of NADH and FADH2. Those are also forms of energy, but really we want ATP. So we want to convert that energy, NADH and FADH2, into ATP. And we do that through something called the electron transport system. And we get a lot more ATP out of it. Okay, so that's kind of the overview. So let's go through each step in a little bit more detail. So here's our glycolysis. So remember, this occurs in the cytosol of your cell. You started off with your 1 glucose, and we just did a bunch of changes to it. Now, you really don't need to know every single step. You need to know that you start with 1 glucose, you go through the process of glycolysis, and you make some energy. How much energy do you get? You get, and I'm going to focus on net gain, you get a net gain of 2 ATPs and 2 NADHs. Now, you really make 4 ATPs. You make 1, 2, and you split here, so this actually happens twice. So you'll make 1, 2, 3, 4. But notice up here, we actually used 1, 2 ATPs. We actually had to put in some energy in the process of getting glycolysis to occur. But again, I'm going to focus on net energy gain. So the net energy gain is 2 ATP, 2 NADHs. Here's our NADHs. Remember, we split here, so it's 2 of everything here. And we end up with 2 pyruvates. So what do we do with the pyruvates? Well, we're doing aerobic metabolism. So focus down here. The 2 pyruvates will then move into the mitochondria, so the inner membrane, so in the matrix of the mitochondria. And the 2 pyruvates will turn into 2 acetyl-CoAs. And we're going to make 2 NADHs because each one makes 1. Now, once you're in the mitochondria, every step, somewhere, you're going to need oxygen. And so notice we create CO2 as a byproduct. This is why it's called aerobic, because we need oxygen. And we make 2 NADHs. So again, we're going to keep track. So then what happens to the 2 acetyl-CoAs? Well, the 2 acetyl-CoAs will go through the citric acid cycle, also known as the Krebs cycle. And for every 1 acetyl-CoA that goes into the Krebs cycle, you get 1, 2, 3 NADHs, 1 ATP, and 1 FADH2. Since 2 goes in, you're going to get 6 NADHs, 2 FADH2s, and 2 ATP. Oxygen is also required in the Krebs cycle, which is why we create CO2 as a byproduct. Now, all the ATPs that we created so far, that's fantastic. We can use it. But the NADH and FADH2s, we can't. So what we're going to do is we're going to take those high energy electrons, that's your NADH and FADH2, and we are going to put it through the electron transport system. Basically what we're going to do is we're going to use NADH and FADH2's energy to pump hydrogen ions from the matrix into the inner membrane space. So here's the outer membrane, inner membrane. We're going to put it into this space. So notice pump, pump, pump hydrogen ions. So what does that do? It creates a high concentration of hydrogen ions in the inner membrane space. Ions want to move from high to low concentration. So these hydrogen ions want to move back into the matrix, and it will. And it'll do that through this guy called the ATP synthase. And as hydrogen ions flow through the ATP synthase, this guy synthesizes ATP. So he uses that kinetic energy to make ATP. And we get a lot of ATP this way. Now, one other thing I want to point out is when those high energy electrons, NADH and FADH2, go through the electron transport chain, you end up with a low energy electron. We want to get rid of this, and we do that by combining it with oxygen to create water. So during ATP production, we actually create water. So let's summarize our energy gain. So in aerobic metabolism, from 1 glucose, when we put it through glycolysis, we get 2 NADHs and a net gain of 2 ATPs. Our 2 pyruvates turn into 2 acetyl-CoAs, and we get our 2 NADHs through that. And in the citric acid cycle, we get 6 NADHs, 2 FADH2s, and 2 ATP. Notice oxygen is required once we go into the mitochondria, which is why we create CO2 as a byproduct. All of the ATPs we created, we use just fine, but the NADH and FADH2s have to go through the electron transport system. Now, how much ATP do we get by these guys going through the electron transport system? Well, look over here. For every NADH that goes through the electron transport chain, we get 2.5 ATP molecules. For every FADH2, we get 1.5 ATP molecules. So in total, we have our 4 ATPs that we created, 1, 2, 1, 2. The 10 NADHs, 6 plus 2 plus 2 is 10. For every NADH, we get 2.5, so we're going to multiply that. For the two FADH2s that we created here, and for every FADH we get 1.5, so 2 times 1.5. And if we do the math, we get a net gain of 32 ATP. So for aerobic metabolism, starting off with 1 glucose, we get a net gain of 32 ATP molecules. Now, what happens if you don't have oxygen? Then we do anaerobic metabolism. Now, for anaerobic metabolism, we can still do glycolysis because glycolysis didn't require oxygen. So we're still going to do glycolysis with our 1 glucose, getting 2 ATPs, 2 NADHs, and our 2 pyruvates. Now in this step, the pyruvates cannot go into the mitochondria because we don't have oxygen to turn it into acetyl-CoA. So instead, the pyruvates turn into lactate. Now this step actually requires NADH. So if we look over in our summary, okay, of anaerobic metabolism, in glycolysis, we made 2 NADHs and 2 ATPs, net gain. But to take our 2 pyruvates and turn it into 2 lactates, we use 2 NADHs. So in total, we don't get any NADHs produced for us to use. Instead, all we get is 2 ATP. So in aerobic metabolism, from 1 glucose, we made 32 ATP molecules. In anaerobic, from 1 glucose, we only made 2. So you see there's a huge difference here. So oxygen is good for you. All right, next, this is an example of just lipid synthesis, and just to remind you, the smooth ER is where this occurs, and here's a triglyceride that's been formed. But I want to focus on protein synthesis, and this will be our last thing. So protein synthesis starts in the nucleus. So in the nucleus, we have our genetic code, our DNA, our double-stranded DNA. Remember our nucleotides, adenine, thymine, guanine, cytosine, and that's how they pair up. And for RNA, just remember that the thymine is replaced with uracil. So our DNA double-stranded molecule will unzip, and then what we're going to do is we are going to transcribe it, basically copy it into RNA. And the RNA is shown here in green. Once we transcribe it, once we take one strand of our DNA and copy it into the RNA that would match, we have our mRNA. M is for messenger RNA. The messenger RNA, because it's single-stranded, can exit the nucleus. And once it exits the nucleus, here it is. It comes in contact with a ribosome. Now remember, we have ribosomes that are free. We have some ribosomes that are fixed into the rough ER. Now here's what the ribosome does. The ribosomes will read your mRNA nucleotides 3 at a time. So here what you're seeing is a guanine, adenine, uracil. Now who pairs with guanine? Cytosine does. Who pairs with adenine? Uracil. Remember, we're talking about RNA. So normally adenine goes with thymine, but because we're talking about RNA, it's going to go with uracil. And so who pairs with uracil? It would be adenine. So what happens is the ribosomes will read the mRNA 3 at a time, 3 nucleotides at a time, and it will essentially call over a tRNA that will fit. And this tRNA shown here carries a unique amino acid. So it brought over this amino acid here. Then the ribosome moves on to read the next 3. So the next RNA sequence is UUC. So the tRNA that comes over is an AAG, and it also carries an amino acid. And as these amino acids come, we know already that we form peptide bonds to bond them together. And that's how we create our primary protein sequence. And we talked about that previously. Our primary protein sequence will then turn into our secondary, tertiary, quaternary, etc. So again, here's our DNA. We will then transcribe DNA into mRNA. mRNA will then get translated into our protein. And that process of translating it involves our ribosome reading it and calling over a tRNA to bring an amino acid. And so here we have a free ribosome doing that. And when a free ribosome does that, the protein that's made stays within the cell. When it's a fixed ribosome, so the rough ER makes the protein, then it will get sent to the Golgi from the rough ER. And remember, the Golgi puts proteins into little secretory vesicles, and when the correct signal comes along, it will then exocytose the contents to the extracellular space. So that is a summary of Chapter 4. Thank you.